So for the second part of our lecture this week, I'll be discussing to you the endomembrane system components. So first, we have the endoplasmic reticulum, or the ER, and I'll be discussing about the information and general functions. So the history and general description of the endoplasmic reticulum was first detected in the 19th century, wherein it has a vague cytoplasmic network seen in stained cells, or the ergastoplasm, and later seen in the emission microscopes by Porter, who renamed it into endoplasmic reticulum. So it is actually divided into two broad categories. So the first one would be the rough and the second one would be the smooth. And both enclosed space, so cytoplasm, divided into cytosolic and luminal cisternal spaces, and contents of the two spaces are quite different. So in fluorescently labeled proteins and lipids, it can diffuse from one type of ER into the other. So it's indicating that their membranes are continuous. So when you say smooth ER or the SIR, it is typically tubular, it has interconnecting pipeline system and curves through the cytoplasm. So hence it's being known as the smooth endoplasmic reticulum. It lacks the association of ribosomes. So when cells are homogenized, it fragments into smooth surfaced vesicles. While on the other hand, for the rough endoplasmic reticulum, it is an extensive organelle with ribosomes attached to it on its cytosolic surface, and it is made mostly of the cisternae, and space inside appears continuous. So different cell types contain varying amounts of either one ER type or other, and it depends on the cell activities being performed. So for the smooth ER functions, they are actually extensively developed in many cells, and its specific proteins vary cell to cell depending on the functions of the cell's smooth endoplasmic reticulum. So it includes the synthesis of steroid hormones in the gonads and adrenal cortex endocrine cells. It also performs detoxification in liver of many organic compounds, whose chronic use can lead to the proliferation in liver cells, and detoxification is carried out by oxygen transferring enzymes. Lastly, it has the function of sequestering calcium ions within cisternal space, and their release triggers specific cell activities. For the rough endoplasmic reticulum functions, predominantly is for exporting or membrane protein synthesis, and organelles of protein secreting are glandular epithelium cells, which are distinctly polarized along the cell-tall axis and reflects flow of secretory products from synthesis to discharge. It was also found that the rough endoplasmic reticulum is a secretory protein synthesis site in the pancreatic acinar cells. So for the synthesis of proteins on membrane-bound versus free ribosomes, so further experiments revealed that polypeptides are synthesized at two distinct locales within the cell. So some proteins are made on the ribosomes attached to the cytosolic surface of the rough endoplasmic reticulum membranes, and the other polypeptides are made on the free ribosomes and then released into the cytosol. So, why are proteins made at different cell sites and how are they identified? So, basically, this has been supported by the signal hypothesis, which earned the Nobel for Medicine in 1999 by Gunther Blobel, David Sabatini, and Bernard Doberstein from the Rockefeller University in the early 1970s. So, their hypothesis suggested that um, and actually demonstrated that the site of the protein synthesis is determined by the information contained in the N-terminal portion of the protein. So some transport into the ER is post-translational, wherein protein is made totally in cytosol and then imported into the endoplasmic reticulum. So signal hypothesis has been substantiated by a large body of experimental evidences afterwards. So this one is the steps, or these are the steps in the synthesis of the secretory, lysosomal, and plant vacuolar protein on the membrane-bound ribosome. So with this one, I'll not be discussing it one by one. Just read up on the different steps. And here is actually the figure showing you the schematic model of the synthesis of the secretory protein on a membrane-bound ribosome of the rough endoplasmic reticulum. So this image is specifically found on page 269 of your book. So just read up on the steps that are involved with this type of synthesis process. Okay. So next, GTP is involved in the secretory protein synthesis. So wherein there are several steps are regulated by its binding or hydrolysis. So G proteins, or the GTP binding proteins, play a key regulatory role in many different cellular processes, together with the SRP and SRP receptors, which are two major interactants in the protein synthesis process, are both G proteins. Okay? So, processing of the newly synthesized proteins in the ER, as nascent polypeptide enters the rough endoplasmic cisterna, it is acted upon by a variety of enzymes located within either the membrane or lumen of the rough endoplasmic reticulum. So the signal peptide on the N-terminus of the nascent polypeptide is removed from most of the nascent proteins by a proteolytic enzyme you call the signal peptidase, and carbohydrates are added to the nascent protein by the enzyme oligosaccharyl transferase. So the RER is a major protein processing plant. To meet its obligations, the rough endoplasmic reticulum lumen is packed with molecular chaperones that recognize and bind to the unfolded or misfolded proteins and give them opportunity to attain their correct native 3D structures. Thus, lumen also contains a number of protein processing enzymes like the protein disulfide isomerases, or PDI. So the ER is ideally constructed for its role as a port of entry for the biosynthetic pathway of the cell. So its membrane provides a large surface area to which many ribosomes can attach, and the ER cisternae lumen provides local environment that favors protein folding and assembly. So the ER cisternae lumen also provides a compartment in which secretory, lysosomal, and plant cell vacuolar proteins can be segregated from the other newly made proteins. So again, the importance of, having ribosomes on the endoplasmic reticulum is that because ribosomes are the ones responsible for, generating proteins inside the cell. Okay? So for the synthesis of the integral membrane proteins on the membrane-bound ribosome, so integral membrane proteins, other than those of the mitochondria and chloroplast, are also synthesized on the membrane-bound ribosomes of the ER. So these membrane proteins are translocated into the ER membrane as they are synthesized using the same machinery used for the synthesis of the secretory and lysosomal proteins. With the use of your X-ray crystallographic studies of translocon, show that translocon have a clam-shaped conformation with a groove or seam along the one side of the wall where the channel might open and close. So this idea has received strong support from in vitro studies in which translocons were given the chance to translocate custom-designed nascent proteins containing test segments of varying hydrophobicity. So single-spanning membrane proteins can have also an orientation with their N-terminus facing either the cytosol or the ER lumen, and eventually the extracellular space. So the most common determinant of membrane protein alignment is the presence of positively charged amino acid residues flanking the cytosolic end of a transmembrane segment. So during membrane protein synthesis, the inner lining of a translocon is thought to orient the nascent polypeptide so that the more positive end faces the cytosol. In multi-spanning proteins, sequential membrane segments have opposite orientations. So for these proteins, every other transmembrane segment has to be rotated 180 degrees before it can exit the translocon. And studies performed with purified components in cell-free systems suggest that a translocon by itself is capable of properly orienting transmembrane segments. It also appears that translocon is more than a simple passageway through the endoplasmic reticulum membrane, and it is a complex machine that can recognize various signal sequences and perform complex mechanical activities. Next, we have the membrane biosynthesis in the endoplasmic reticulum. So membranes thought to arise only from pre-existing membranes. And as we all know, membranes grow as a newly made proteins and lipids are inserted into existing membranes in the ER. Each compartment has actually unique membranes, and the cell membranes are asymmetric, which has two phospholipid layers or leaflets that have different compositions. So in this image or in this figure, you will show the maintenance of the membrane asymmetry. So you have here your rough endoplasmic reticulum. You also have the different proteins, the Golgi complex, the vesicles, and of course, the plasma membrane. So this part shows you the exterior side, and this is the cytosolic part of your cell. Okay? So the synthesis of the membrane lipids, most membrane lipids are produced entirely in the ER membrane with the following exceptions. We have the sphingomyelin and glycolipids, where the synthesis of which starts in the ER and is completed in the Golgi complex. Okay? So some unique mitochondrial or chloroplast membrane lipids made by the enzymes is in those of the membranes. So again, not all of the synthesis happens in a particular organ or in a particular organelle within the cell. So some would actually kickstart from one organelle and then eventually would complete its process to another organelle. Okay, so phospholipids are made by integral ER membrane enzymes whose active sites are or actually facing the cytosol. Membranes of different organelles have markedly different lipid compositions, and what factors do actually contribute these changes? So most proteins made on the RER are glycosylated and thus become glycoproteins. Whether integral proteins of the membrane are soluble, lysosomal, or vacuolar enzymes or parts of the ECM. So we have carbohydrate groups which have key roles in the function of many glycoproteins, and they also aid in the proper folding protein to which they are attached. So examples would be, or an example would be the binding sites in their interactions with other macromolecules. And then, how is oligosaccharide sugar sequence assembled? So they are catalyzed by a family of membrane-bound enzymes you call your glycosyl transferases. So carbohydrate chains are attached to the protein N linkages, usually asparagine N atom, or O linkages to the serine or threonine O, or collagen hydroxylysine residue of both soluble and integral membrane proteins. So these oligosaccharides in the glycosylation in the RER differ in average size, sugar composition, and path of synthesis, and also share properties like their high specificity. N-linked basal chain segment is assembled on lipid carrier and not protein, then transferred as a block to specific asparagine residues of polypeptide as it enters the RER by oligosaccharyl transferase. So this image shows you the steps in the synthesis of the core portion of an N-linked oligosaccharide in the endoplasmic reticulum. So again, we will not be dealing in this or more complex types of processes, but I would just like you to read most of the or understand basically the process that are involved in this synthesis. So this image is found or can be located in page 273 of your book. Okay? So next, for the glycosylation, mutations that lead to the total absence of any glycosylation cause death of embryos prior to implantation. So mutations leading to partial glycosylation pathway disruption in ER also cause serious inherited disorders affecting nearly every organ system. So these diseases are called your congenital diseases of glycosylation, or CDGs, and they are usually identified through blood tests that detect abnormal glycosylation of the serum proteins. So one of the examples of these protein is the CDG1B, which can be managed through a remarkably simple treatment. Okay? So some oligosaccharides, especially in lower eukaryotes, are simply the core, but they tend to diversify in more complex organisms. So after block is added, core is modified first in the ER with enzymatic removal of two of three terminal glucose residues by the glucosidases, and this sets the stage for an important event in a newly made glycoprotein's life. So if folding is incomplete or if protein is misfolded, it is recognized and bound by the monitoring enzyme GT. Okay? So, mechanisms that ensure destruction of misfolded proteins. So, again, whenever that you have, your misfolded proteins, it usually gives you the idea that most likely there will be mutations that will happen on those produced proteins. So, misfolded proteins are not destroyed in the ER but are instead transported into the cytosol by dislocation process. So it remains unclear whether misfolded proteins are dislocated back into the cytosol through translocons that brought them into the ER or by way of a separate dislocation channel of certain identity. So this is not yet very clear how these misfolded proteins are being evicted or are being transferred from the site of where they were created up until they are, removed from the cell itself. So once in the cytosol, misfolded proteins are destroyed in the what you call your proteosomes, which are protein degrading machines, and this process ensures that aberrant proteins are not transported to other parts of the cell. Okay? Because again, once misfolded proteins are transferred to other parts of the cell and they are being utilized for a certain process, of course, it would give a negative result on the process itself. Okay? So sometimes misfolded proteins can be generated in ER at a rate faster than they can be exported to the cytoplasm. So the accumulation of misfolded proteins, which is potentially lethal situation, triggers a comprehensive plan of action within the cells known as the unfolded protein response, or UPR. So the ER contains sensors that monitor the concentration of unfolded or misfolded proteins in the ER lumen, and one proposal suggests that the sensors are normally kept in an inactive state by molecular chaperones. So among the genes expressed are genes that encode the ER-based molecular chaperones that can help misfolded proteins reach the native state, and proteins involved in the transport of the proteins out of the ER, and the proteins involved in the selective destruction of abnormal proteins. Okay? So the UPR, or the unfolded protein response, is more than cell survival mechanism, and it includes the activation of cell death pathway. So from the ER to the Golgi complex, let's talk about the first step in the vesicular transport. So the exit sites of the RER cisternae are typically smooth surface or devoid of ribosomes and serve as place where the first transport vesicles in the biosynthetic pathway are formed. So trip to the ER to the Golgi apparatus has been visualized in living cells by tagging secretory proteins with green fluorescent protein. So as you remember, there are, this GFP has been very helpful when it comes to tracing the pathways of these different proteins. Okay? So after budding from ER membrane, transport vesicles are seen to fuse to each other to form larger vesicles and interconnected tubules in a region between the ER and the Golgi complex. So this region is called the ergic or the endoplasmic reticulum Golgi intermediate compartment, and the vesicular tubular clusters that form there are called VTCs. Okay? So once formed, VTCs move farther away from the ER towards the Golgi complex, and other studies indicate that this movement occurs along tracts composed of the microtubules. So the Golgi complex is actually discovered by Camillo Golgi, an Italian biologist, in 1898. He is the inventor of new types of staining procedures that he hoped might reveal the organization of nerve cells within the central nervous system. So one stain used for the solution is the silver nitrate that has been applied to tissue that had been soaked in osmium and bichromate. Existence confirmed beyond a reasonable doubt that when it was clearly identified in unfixed freeze-fractured cells, it was no artifact. So characteristic morphology of the Golgi complex is actually flattened, disc-like membranous cisternae with dilated rims and associated vesicles and tubules. So smooth membranes are also found with smooth microsomes. So the cisternae is typically around 0.5 to 1 micrometer in diameter, arranged in orderly stack like pancakes. They are curved, resembling a shallow bowl. Individual Golgi stacks often interconnected to form ribbon-like complex. So usually, less than 8 cisternae are present per stack, but may have a few to several 1,000 distinct stacks or cells depending on the cell type. Okay? So the Golgi complex has a polarized Golgi or Golgi cisternae. So it has a cis phase, which is the entry phase closest to the ER, and the trans phase, which is used as the exit at the opposite end of the stack, closer to the plasma membrane. So membranous elements of the Golgi complex may be supported mechanically by a peripheral membrane skeleton or scaffold composed of a variety of proteins. So the Golgi complex composition is not uniform from cis to trans end. It is polarized. Differences in the composition of the membrane compartments reflects primary processing plant role, and as they move along the stack, proteins originally synthesized in the RER are sequentially modified in specific ways. So glycosylation in the Golgi complex. This is actually the synthesis site of most cells' complex polysaccharides, including animal ECMs, GAGs, plant cell wall pectins, and hemicelluloses. So this is also where the key role in glycoprotein glycolipid protein assembly happens. So in the ER, glucose residues had just been removed from the ends of the core oligosaccharide of N-linked protein chains, and in the Golgi, as in ER or as in the rough endoplasmic reticulum, sequences in which sugars are inserted into oligosaccharides is determined by spatial arrangement of specific glycosyl transferases that contact new proteins as they pass through. So unlike N-linked oligosaccharides, whose synthesis starts in ER, those attached to proteins by O linkages are assembled wholly within the Golgi complex. So the vesicular transport within the Golgi are happening actually in or is supported by two contrasting theories. So first is the cisternal maturation model, which was accepted up to the mid 1980s. It was accepted that cisternae were transient structures that form at cis phase by ER or ergic vesicle fusion, traveled to trans phase, and altered along the way. However, the new model was favored in the mid-1980s until the late 1990s, wherein Cisternae of the Golgi stack remain in place as stable compartments held together by protein scaffold known as the vesicular transport model. So, acceptance of the vesicular transport model based largely on the following observations. So, each of the various Golgi cisternae of stack has distinct resident enzyme population. And how could this various cisternae have such different properties if each gave rise to the next in line as stated by other models? So this has been answered by James Rothman et al. By Stanford from Stanford in 1983, wherein large numbers of vesicles are seen in an electron micrograph to bud from the rims of the Golgi cisternae. So both models still have proponents, but consensus has shifted in the past few years back to cisternal maturation model, and several major reasons summarized below. So the cisternal maturation model envisions a highly dynamic Golgi complex in which major elements of the organelle, the cisternae, are continually being formed at the cis phase and dispersed at the trans phase. So certain materials that are produced in ER and then travel through Golgi complex can be shown to stay within Golgi cisternae and never appear within the Golgi associated transport vesicles. Until the mid-1990s, it was assumed that transport vesicles always moved in forward or in the process of anterograde direction from cis origin to trans destination, but with new evidence that says that some move in backward or retrograde direction from trans donor to cis acceptor membrane. So, revised cisternal maturation model acknowledges a role for transport vesicles, which have been clearly shown to bud from the Golgi membranes. So, in this model, transport vesicles do not shuttle cargo in anterograde direction, but instead carry resident Golgi enzymes in retrograde direction. So, in another experiment, treated gold-labeled antibodies that bind to a Golgi resident protein. It was found in both the cisternae and associated vesicles. So the revised cisternal maturation model explains how different Golgi cisternae in stack can have a unique identity, and some prominent researchers still argue that cargo can be carried by transport vesicles between Golgi cisternae in an anterograde direction, so the matter is not yet settled. However, it is more, or they are, or the model that's more acceptable is the maturation model. Okay? Cisternal maturation model. So the types of vesicle transport and their function. So materials carried between membrane compartments by vesicles or other types of membrane-bound carriers which bud from donor membranes and fuse with acceptor membranes. So most budding vesicles covered on cytosolic surface by fuzzy electron-dense layer, and protein coats have at least two distinct functions. And then components selected for transport can include cargo to be transported. It can be secretory, lysosomal, and membrane proteins. And of course, the machinery required to target and dock the vesicle to an acceptor membrane. So protein coats are able to make these selections by virtue of specific affinity for the cytosolic tails of integral proteins that reside in the donor membrane. Vesicle membrane phospholipids also play an important role in the selection. So, several distinct classes of coated vesicles have been identified and distinguished by the proteins that make up the coat, their appearance in the emission microscope, and their role in cell trafficking. And there are, three best vesicle transports, or three vesicles that are best studied. Okay, so here we have the first one. This is actually not one of the three that are commonly studied. So we have the TGN, to the endosome, lysosome, and plant vacuoles. The three that are best studied are the CAP2, coated vesicles, which move materials towards the ER to the ergic and Golgi Complex. So COP is the acronym for coat proteins. And then we have the COP1, coated vesicles, they move materials in retrograde direction from the ergic and Golgi stack backward towards the ER and the clathrine-coated vesicles. So for the COP2-coated vesicles, they are the most recently discovered and mediate the first leg of the journey through the biosynthetic pathway from the ER to the ergic and the CGN. So, COP2 coat contains a number of proteins first found in the mutant yeast cells that could not transport materials from ER to Golgi, and they are homologous proteins found in coats of vesicles budding from mammalian cell ER. So, the antibodies to COP2 coat proteins block vesicle budding from ER membranes but have no effect on the movement of the cargo and other stages in the secretory pathway. So, COP2 vesicles are thought to be able to select and concentrate certain components that they transport. So, they are selectively capturing um, these integral membrane proteins because they act specifically with COP2 proteins of vesicle coat, and several types of membrane proteins are included in this group. For example, you have the ergic 53 mutations have been linked to an inherited bleeding disorder, and people with the disorder fail to secrete certain coagulation factors that promote blood clotting. So, interactions between the membrane proteins like ergic 53 and the COP2 coat is mediated by signal sequences in the cytosolic tails of the membrane proteins. So, ergic 53, for example, is recognized by two neighboring phenylalanines in each cytosolic tail, and other types of the soluble cargo are not selected at this stage and are present at the same concentration in the budding vesicles as in ER lumen. So, among the COP2 proteins is a small G protein, what we know as the SAR1. It is recruited specifically to ER membrane. And like other G proteins, SAR1 plays a regulatory role, and it starts the vesicle formation and regulates vesicle coat assembly. So, here, actually, or this slide actually shows you the stages on how this SAR1 is being recruited to the ER membrane and to be able to produce, the COP2 coated vesicles. So just read up on this one, and I will no longer go in details with this. So COP1 coated vesicles were first identified in experiments where cells were treated with GTP analogs that cannot be hydrolyzed, unlike your GTP. So in presence of these analogs, COP1 coated vesicles accumulated within the cell, and they could be isolated from the homogenized cells by density gradient centrifugation. And then COP1 coated vesicles have been most clearly implicated in the retrograde transport of the proteins, including some movements, and whether or not COP1 coated vesicles are involved in the anterograde and or retrograde transport between Golgi cisternae and remains a matter of controversy. So for the COP1 coated vesicles, transporting escape proteins back to the ER. So retaining and retrieving resident ER proteins is very important. And, vesicles continually bud from the membrane compartments. And the question is that how each compartment retains its unique composition. So, what determines whether a particular ER membrane protein stays in the ER or goes into the Golgi complex? So, studies suggest proteins are maintained in an organelle by a combination of two mechanisms. The first one is the retention of resident molecules that are excluded from the transport vesicles, and the second one would be the retrieval of escape molecules back to the compartment in which they are or which they normally reside. So this mechanisms of retention in particular membrane is actually not well understood. But here we have a figure that would show you the retrieving process in the ER proteins. Okay? So retrieval of escape protein is better understood compared to the um, uh, of the retaining of these resident molecules or proteins. And proteins that normally reside in the ER have short amino acid sequences at the C terminus that serve as retrieval sequences. So it ensures the return to the ER if they are carried forward accidentally in transport vesicle to Ergic or the Golgi complex. So retrieval of escaped ER proteins from these compartments is accomplished by specific receptors that capture the molecules and return them to the ER in COP1 coated vesicles. So soluble proteins of the ER lumen typically possess the retrieval signal that includes the lysine, asparagine, glutamine, and lutein, as or usually known as the KDEL in nomenclature. So the ER integral membrane proteins like the SRP receptor have different retrieval signal at C terminus, and it binds to COP1 coat and ensures its return. Okay? So each biosynthetic pathway compartment may have its own unique retrieval signals, which explains the maintenance of unique protein complements in each one despite constant in and out vesicle movement. So sorting of the lysosomal proteins at the TGN. So how does particular protein synthesize in ER get targeted toward a particular cellular destination? So cell must be able to distinguish among the various protein it manufactures. Okay, so that is a, a, a specific requirement. Okay, and then protein sorting occurs in the last of the Golgi compartments, known as the trans-Golgi network, which functions as a major branch point in the movement of materials along the secretory pathway. So lysosomal protein sorting and transport are made on the membrane-bound RER ribosomes, carried to the cis-Golgi cisternae with other protein types. And this is the best, this is best understood in post-Golgi pathway for lysosomal enzymes. So once in Golgi cisternae, soluble lysosomal enzymes recognized by enzymes catalyzing two-step addition of phosphate group to certain N-linked protein chain monosugars. And then lysosomal enzymes with mannose-6-phosphate signals are recognized and captured by the mannose-6-phosphate receptors. Or this is what you call your MRPs, which are integral membrane proteins that span the Golgi membranes. So, lysosomal enzymes are transported from the TGN in clathrin-coated vesicles, and coats of the vesicles actually contain an outer honeycomb-like lattice composed of the protein clathrin, which forms a structural scaffold, and an inner shell made of protein adapters that cover the vesicle membrane surface facing the cytosol. In molecular biology, an adapter is a molecule that physically links two different types of materials. And lysosomal enzymes are exported from the TGN by a family of adapter proteins called as GGAs. Okay? So, as in COP1 or COP2 vesicle formation, clathrin-coated vesicle production starts with the recruitment to the membrane of small GTP-binding protein or ARF1, which sets the stage for binding of other coat proteins. So once the vesicle has budded from the TGN, the clathrin coat is lost, and the uncoated vesicle proceeds to its destination, which may be an endosome, lysosome, or a plant vacuole. So once the vesicle reaches its destination organelle, the MPRs dissociate from the lysosomal enzymes and return to the TGN for another round of lysosomal enzyme transport. So next, let's go to the sorting and transport of non-lysosomal proteins. So it is good to remember that membrane proteins are destined for plasma membrane, and secretory proteins are destined for export from the cell, are also transported from TGN, but the mechanisms are poorly understood. But with recent models, membranous carriers are produced as the TGN fragments into vesicles and tubules of various sizes, and this fits with the cisternal maturation model. So the targeted delivery of the integral proteins to The plasma membrane appears to be based largely on sorting signals in the cytoplasmic domains of the membrane proteins. So as in polarized cells, membrane proteins destined to reside in the apical portions of the plasma membrane contain different sorting signals from those destined from the lateral or basal portions. Hence, plasma membranes of non-polarized cells may not require special sorting signals. So, for the targeting vesicles to a particular compartment, so vesicle fusion requires specific interactions between different membranes. So, vesicles from the ER fuse with ergic or cis Golgi network and not with trans-cisterna. Selective fusion occurs and is one factor that helps ensure a highly directed flow through the membranous compartments of the cell. So for you to be able to understand the targeted or targeting transport vesicles to its particular target membranes, you may want to check figure 8.31 of your book from page 289. Okay, so the way in which cells target vesicles a specific compartment is not well understood, but vesicles are thought to have specific proteins in their membranes governing their movements and fusion potential. So the summary of the steps between vesicle budding and vesicle fusion is needed to understand the nature of the proteins in the vesicle membranes controlling vesicle movement and fusion. So summary of the steps between vesicles budding and fusion. Movement of vesicle towards a specific target compartment must sometimes move large distances through cytoplasm before reaching its eventual target, probably directed by the signals coming from your microtubules. And these microtubules act like railroad tracks carrying cargo containers along a defined pathway to a predetermined destination. So tethering vesicles to the target compartment has been explained through the microscope studies indicating that vesicles are often tethered to a presumed target compartment like Golgi cisterna by extended fibrous proteins. So tethering may be an early stage in the process of vesicle fusion that requires specificity between vesicles and target compartments, and much of this specificity may be conferred by a family of small GTP binding proteins, for example, your RABs. So next is the docking of vesicle to target compartments. At some point during the process leading to the vesicle fusion, membranes of vesicle and targeting compartment become tightly opposed to one another. So this is a result of the interaction between the cytosolic regions of the integral proteins of the two membranes. So snares are divided functionally into two categories. We have the V snares, which are incorporated into the transport vesicle membranes during budding, and the T snares, which are located in the target compartment membranes. So they are best studied. The best studied snares are those that mediate docking of synaptic vesicles with the presynaptic membrane during the regulated release of the neurotransmitters. And interestingly, the snares of the synaptic vesicle and presynaptic membranes are targets of two of the most potent bacterial toxins and those of that responsible for botulism and tetanus. So, fusion between the vesicle and target membranes when artificial lipid vesicles or liposomes containing purified T snares are mixed with liposomes containing a purified V snare. The two types of vesicles fuse with one another but not themselves. So one view regarding the regulated secretion of the neurotransmitter molecules is how specificity of this interaction being determined. So the current consensus is that the ability of a particular vesicle and target membrane to fuse is determined by a specific combination of the interacting proteins. So next, for exocytosis. So this is the terminal stage of the secretion. So it is best studied, or the best studied examples of the vesicle fusion are regulated fusion of the secretory or synaptic vesicles with the plasma membrane. So in these cases, membrane fusion produces opening through which vesicle or granule contents are released into the extracellular space. And the process of the membrane fusion and content discharge is what you call your exocytosis. It is usually triggered by a local increase in calcium ion concentration. Okay? So, the steps in the exocytosis is actually not well understood, but cell and vesicle membrane contact mediated by fusion proteins within and on membrane surface, and where proteins thought to create close-range contact between membranes destined to interact and fuse. And contact between the cell and the vesicle membranes may lead to the formation of small protein line fusion pore that rapidly dilates to form opening for discharge. So lysosomes. So for the lysosomes, morphology and contents, they typically contain at least 50 different hydrolytic enzymes made in the RER and targeted for lysosomes. So lysosomes are an animal cell's digestive organelle. So the lysosome enzymes taken toward can hydrolyze virtually every type of biological macromolecule, resulting in low molecular weight products that can be transported across the lysosomal membrane into the cytosol. All of the enzymes have pH optimum at acid pH or acidic hydrolases, which is suited to the low pH of the lysosomal compartment, and the lysosome interior pH is around 4.6. So the lysosomal morphology is neither distinctive nor uniform, although they house a predictable collection of enzymes, and they are dynamic organelles capable of rapid fusion and fission. So here shows you um, the lysosomes, and of course a summary of the autophagic pathway. So this is how your different vesicles do process to be able to finish an exocytosis process. Okay, so for the lysosomal functions, materials brought into the cell from extracellular environment are enzymatically broken down first, and it results to nutrients cross-membrane into the cytosol, and this is actually the best studied function of all. So it also has the function of fertilization, wherein the sperm head contains specialized lysosome or the acrosome, which contains typical lysosomal enzymes. Unusual because lysosomal enzymes are active outside the cell. But in fertilization, especially or particularly in the sperm head, they are contained there. Okay? So it is also very useful in organelle turnover or autophagy. It is regulated destruction of the cell's own organelles and their replacement. So in recent years, autophagy has also been shown to help protect an organism against intracellular threats, ranging from abnormal protein aggregates to invading bacteria. And once digestive process in autophagolysosome is completed, organelle is called a residual body. Next, let's go now to the plant vacuoles. So this is a single membrane-bound fluid-filled central vacuole that occupies up to 90% of the cell volume, and they have a wide spectrum of essential functions. So it includes the temporary storehouse for many cell solutes and macromolecules. It may also store host of toxic compounds. It also generates high trigger pressure that pushes outward against cell wall and maintains cell shape. And these are the sites of intracellular digestion similar to animal lysosomes. And however, lysosomes are absent in the plants. Okay? So here it shows you an image of how the plant cell vacuoles look like. So if this is the cell wall, and then you see the chloroplast in green, the vacuoles are found, or the vacuole is found in between these structures. Okay? So for the endocytic pathway, this is for the background, the overview. This is moving membrane and materials into the cell interior. So cells must take in materials that are too large to penetrate the membrane regardless of its permeability properties, and recycle proteins that reside in plasma membrane to cells' internal compartments. So both of these requirements are met by the endocytic pathway, in which segments of the plasma membrane invaginate to form cytoplasmic vesicles that are transported to cell interior. So two separate processes of uptake extracellular materials into cytoplasmic vesicles occur by different mechanisms. So the first one would be the endocytosis, and then the second one would be phagocytosis. So terminology has exchanged in recent years. In 1963, C. de Duve or C. de Duve introduced the term endocytosis to include ingestion of particles, specifically phagocytosis, and uptake of fluid and solutes, the pinocytosis. Okay, so again, for cell eating, it's the phagocytosis, and for fluid intake, it's or cell drinking, it would be the pinocytosis. So endocytosis is the uptake of fluid, dissolved solutes, suspended macromolecules, and is divided into two broad categories. The first one is the bulk phase, and the second is the receptor-mediated endocytosis. So for the bulk phase, it is also known as the pinocytosis. It is a type of non-specific uptake of extracellular fluids without the recognition by the membrane. And for the receptor-mediated endocytosis, or RME, it brings about the uptake of specific extracellular macromolecules that bind to receptors on external plasma membrane surface. So the rate of both processes can be remarkably rapid. It can take up to half membrane surface and can be internalized in as little as 30 minutes. So despite rapid inward movement of plasma membrane, there is no shrinkage of the cell surface, nor is there any immediate need for synthesis of new membrane components. So membrane is simply cycled between surface and cell interior, so that membrane is added to surface as fast as it is removed, and exocytosis rate equals to that of the endocytosis. So remember that the membrane is always being recycled. So for the receptor-mediated endocytosis and the role of coated pits, so RME provides for selective and efficient uptake of macromolecules that may be present at relatively low concentrations in the extracellular fluid. So cells have receptors for the uptake of many different types of ligands, and the coated pits are membrane surface sites that are intended and covered on cytoplasmic phase by bristly electron-dense protein layer containing clathrine and adaptors. So the structure of the coat is as viewed from its cytoplasmic surface, as bristly coat appears to consist of network of polygons, sometimes hexagons and pentagons, resembling a honeycomb, and explains the formation of the coat. So more likely has a geometric construction and is derived from the structure of its clathrine building blocks. So triskelions within the clathrine scaffold of a coated vesicle are found in an overlapping arrangement. So if you're familiar with the triskelion logo, that's how this coated pits look like. So for you to check, or for you to visualize the molecular organization of a coated vesicle, please check on figure 8.40 or 8.40 of page 295 from your book. Okay? So, like clathrine-coated vesicles budding from TGN, coated vesicles formed during endocytosis also contain a layer of adaptors situated between clathrine lattice and vesicle surface facing the cytosol. So, the best studied adapter operating in connection with the clathrine-mediated endocytosis is AP2. So unlike GGA adapters used at TGN, AP2 adapters incorporated into vesicles budding from cell membrane have multiple subunits with different functions. So unlike cup 1 and cup 2 vesicles, which have relatively simple construction, clathrin-coated vesicles have upwards of 2 dozen different accessory proteins, forming a very dynamic network of interacting molecules. Okay? So those are the differences. Now, dynamine is a large GTP binding protein that is required for the release of a clathrin-coated vesicle from the membrane on which it forms. So dynamine self-assembles into a helical collar around the neck of an invaginated coated pith, just beneath or just before it pinches off the membrane. So hydrolysis of the bound GTP by the polymerized dynamine molecules is thought to induce conformational change in the dynamine helix that serves coated vesicles from the plasma membrane. Within a minute of its formation, the coated endocytic vesicles loses its clathrin coat and becomes a smooth surface vesicle that enters the endocytic pathway. So molecules taken into a cell by endocytosis are routed through a well-defined endocytic pathway. So there are two different types of receptors. The first one is the housekeeping receptors. They are responsible for the uptake of materials that will be used by the cell, and the best studied examples are transferrin and LDL receptors. So they mediate the delivery to cells of iron and cholesterol respectively. And then next is the signaling receptors, which are responsible for binding extracellular ligands that carry messages that change cell activities. And these ligands do not actually enter the cell, like the hormones like insulin, growth factors like EGF. So endocytic pathway begins with a dynamic network of tubules and vesicles known collectively as your endosomes. So endosome lumen fluid is acidified due to the activity of endosome membrane, hydrogen ATPase or hydrogen pump. So endosomes are divided into two distinct classes. So they are distinguished from one another on the basis of buoyant density, which allows them to be isolated in different fractions on the density gradient, and of course the pH and then the protein composition. So late endosomes may contain considerable amounts of internal membrane that arises from inward invaginations of the boundary membrane. Receptors taken up by endocytosis are transported in endocytic vesicles to an early endosome, which serves as a sorting station that directs different types of receptors and ligands along different pathways. So housekeeping receptors dissociate from their bound ligands in the acidified endosomal environment, and in contrast, released ligands become concentrated into a sorting compartment before being dispatched to a late endosome and ultimately to a lysosome where final processing occurs. Signaling receptors previously marked with ubiquitin tags do not recycle back to the membrane, but instead are sent on the late endosomes and lysosomes where they will ultimately be destroyed. So steps along the endocytic pathway from an early endosome to a lysosome have been described in various ways by different researchers working on different cells. So several reports say that the transfer of materials from early to late endosomes occur by the means of specialized carrier vesicles, often referred to as multivesicular bodies or MVBs. And molecules that travel along endocytic pathway in the late endosome are ultimately directed to a lysosome and the terminal compartment of the endocytic pathway. This movement occurs by two major routes. Okay? So once in a lysosome, membrane receptors and other macromolecules are destroyed, but transported materials like cholesterol are typically processed for the delivery of the cytosol. So for you to further understand the endocytic pathway, the figure 8.43 from your book would explain the pathway itself, which is found on page 301. So next, we have the LDLs and the cholesterol metabolism. So first, and best studied example of the receptor-mediated endocytosis is the mechanism that provides animal cells with exogenous cholesterol. So cholesterol is a hydrophobic molecule which serves as a precursor of the steroid hormones and an essential part of the plasma membrane in animals, and it can't be free in the blood. So cholesterol is transported through blood as part of the huge lipoprotein complexes, and LDL receptors found mostly in the liver cells are transported to the cell's plasma membrane where they wait for the LDL or the low density lipoproteins. So LDL receptors are concentrated in coated pits even in the absence of ligand. Thus, they are in plasma membrane and ready to take up blood-borne lipoproteins if they should become available. And people with a rare inherited disorder like the Niemann-Pick type C disease lack one of the proteins required to transfer cholesterol out of the lysosomes. So the image showing here, figure 8.44, is an LDL cholesterol. As you see, it has apolipoproteins, phospholipids, and esterified cholesterols and esterified cholesterols making it up. Okay? So LDL and atherosclerosis, or the narrowing of the major arteries. So LDL blood vessels or blood levels have been related strongly to the development of atherosclerosis. So recent studies suggest that atherosclerosis results from a chronic inflammatory response that is initiated by the deposition of LDL within the inner walls of the vessels. And LDL lowering drugs like statins, pravastatin or lovastatin inhibit a key role or key cholesterol synthesis enzyme, HMG CoA or CoA reductase lowers blood cholesterol and heart attack frequency. So the model of the atherosclerotic plaque formation is shown here on this slide. So HDLs or the high density lipoproteins in addition to LDLs transport cholesterol in the blood. So they are similar in constructions but have different proteins, which specifically is the apolipoprotein A1, and play different physiological role in the body. So LDL primarily carries cholesterol molecules from the liver where they are synthesized and packed through blood to the body cells. On the other hand, HDL carries excess cholesterol from body cells membrane to the liver. So while high blood LDL levels lead to heart disease, high blood HDL levels are associated with lowered risk, good for the heart. It seems to remove cholesterol from the blood, but things are not this actually simple. Okay? So next, for phagocytosis or the cell eating, uptake of relatively large particulate matter, more than 0.5 micrometers in diameter. It has extensive or in a few cell types specialized for uptake of particulate matter from environment and delivery to lysosomes. So single-celled heterotrophs like your amoebas and ciliates make their livelihood this way. They trap food particles and smaller organisms and enclose them within folds of plasma membrane engulfing food particles. So in most animals, phagocytosis by certain cells is protective mechanism rather than mode of feeding. Okay? So a proteomic study of macrophage phagosomes revealed the presence of a surprisingly large number of proteins in the seemingly simple membrane-bound vacuoles. So among the proteins found in phagosome membrane were a number of species characteristics of the ER, including the chaperone calnexin, and it was found that most of the phagosome membrane content of macrophage is actually derived from the ER rather than the plasma membrane. And phagocytosis is driven by actin-containing microfilament contractile activities underlying the cell membrane. So not all bacteria ingested by the phagocytic cells are destroyed. In fact, some species hijack phagocytic machinery to promote their own survival in the body. For example, we have the Mycobacterium tuberculosis, which are taken into macrophage cytoplasm by phagocytosis, and phagosome doesn't fuse to lysosome. So bacterium inhibits fusion that would kill it and multiplies within the cell. Next, we have the Coxiella burnetii, which actually causes the fever. Its phagosome fuses with the lysosome, but neither acid, pH, nor the lysosomal enzymes can destroy it. And then next, we have the Listeria monocytogenes. It causes meningitis. It produces proteins that destroy lysosomal membrane integrity, so bacterium can escape into the cell cytosol. Next, for post-translational uptake of proteins, we have the organelle peroxisomes. So, protein trafficking in cell is governed by sorting signals, which are secreted protein signal peptides or mannose phosphate groups of lysosomal enzymes. Receptors that recognize these signals and deliver the protein containing them to the proper compartment. So, four of the cell's major organelles like nucleus, mitochondria, chloroplast, and peroxisomes import proteins through one or more outer boundary membranes. So proteins imported by these organelles contain amino acid sequences that serves as addresses that is being recognized by receptors at organelles' outer membrane as with RER. Unlike RER, which usually imports proteins, translationally, proteins of these organelles are imported post-translationally. So uptake of the proteins into the peroxisomes are very simple. So organelles with only two subcompartments in which an imported protein can be replaced. It has a boundary membrane and internal matrix. So proteins destined for peroxisome possess peroxisomal targeting signal, either a PTS for a peroxisomal matrix protein or an MPTS for a peroxisomal membrane protein. So unlike mitochondria and chloroplast, whose imported proteins must assume an unfolded state, peroxisomes can somehow import peroxisomal matrix proteins in their native folded conformation. So next, for the mitochondria have four subcompartments into which proteins can be delivered. You have the outer mitochondrial membrane, the inner mitochondrial membrane, the intermembrane space, and the matrix. So the mitochondria synthesize a few of their own integral membrane polypeptides, but the vast majority of the organelle's proteins are encoded by nuclear genome. These proteins are synthesized in the cytosol and imported post-translationally, and proteins of the mitochondrial matrix and inner mitochondrial membrane can make up the vast majority of the proteins targeted to mitochondria, so discussion is restricted to them. As with peroxisomal proteins and those of other compartments, mitochondrial proteins contain signal sequences that target them to their home base. So mitochondrial matrix proteins have targeting sequences you call your pre-sequences, which are found at the molecule's N-terminus, and it includes a number of positively charged residues that lie on one face of an extended alpha helix. In contrast, most proteins destined for IMM contain several internal sequences that remain as part of the molecule. And then, before a protein can enter a mitochondrion, several events are thought to take place. Protein must be presented into a mitochondrion in relatively extended or unfolded state, and several different molecular chaperones are implicated in preparing polypeptides for mitochondrial uptake, including ones that specifically direct mitochondrial proteins to cytosolic outer membrane surface, and others unfold polypeptides and prevent their aggregation. So, the outer membrane contains a protein import complex, or the TOM complex, which includes a receptor that recognizes and binds mitochondrial proteins and a protein line channel. So, unfolded polypeptides are translocated through the outer mitochondrial membrane via the protein line channel, and proteins destined for the inner mitochondrial membrane or matrix must pass through the intermembrane space and engage a second protein import complex located in the inner mitochondrial membrane you call the TIM complex or TIM complex. So the internal or the inner mitochondrial membrane contains two major TIM complexes. So the movement into the matrix powered by the electrical potentials across IMM acting on the positive charge targeting signals. So for the importing proteins through the mitochondrion, you may check figure 8.47, page 305 of your book. So as polypeptide enters aqueous matrix, it interacts with mitochondrial chaperones that mediate the entry. Two mechanisms explain the general chaperone role in the protein movement across membranes. So according to one view, chaperones act as force-generating motors that use energy from ATP hydrolysis to pull the unfolded polypeptide through the translocation pore. And then the alternate view is or states that chaperones aid in polypeptide diffusion across the membrane. So this is a random process in which a molecule can move in any available direction. And recent evidences suggest that both of the above mechanisms of chaperone action are probably used and act cooperatively. Next, for the chloroplast. So the chloroplast have 6 subcompartments into which proteins can be delivered. First, you have the inner envelope membrane, this part. You have the outer envelope membrane, and then the intervening intermembrane space, this one. You also have the stroma, the thylakoid membrane, and then the thylakoid lumen. Okay? So in the thylakoid membranes, it does have the thylakoid lumen, the stroma lamellae, and then the grana. Okay? So chloroplast and mitochondrial import mechanisms exhibit many similarities, although their translocation machinery have evolved independently as in the mitochondria. So vast majority of the chloroplast proteins are imported from the cytosol, and the outer and inner envelope membranes contain in these distinct translocation complexes you call the TOC and TIC complexes respectively that work together during the import. So chaperones aid in the unfolding of the polypeptides in the cytosol, enfolding of the proteins in the chloroplast, and proteins destined for the chloroplast are synthesized with a removable N-terminal sequence they term as the transit peptide. So, transit peptide does more than simply targeting a polypeptide to a chloroplast, but it also provides an address that localizes the polypeptide to one several possible subcompartments within the organelle. So, all proteins translocated through the chloroplast envelope contain a stroma targeting domain as part of their transit peptide. This guarantees that the polypeptide will enter the stroma. And then those polypeptides that belong in a thylakoid membrane or thylakoid lumen bear an additional segment in their transit peptide you call the thylakoid transfer domain that indicates entry into the thylakoids. So several distinct pathways have been identified by which proteins are either inserted into the thylakoid membrane or translocated in the thylakoid lumen. So that actually ends my lecture, the second lecture for this week. So for your reference, you may want to read CARP, the Cell and Molecular Biology Concepts and Experiments, 8th edition. So other images were actually lifted from google.com. So if you have further questions, let me know and I'll be more than willing to answer them. Thank you.